Sairam, welcome back to the third session of the fundamental unit of life, that's the cell. Well, I think you went to all the test your skills, you tested your skills, and you came to know where you stand, and also you must have got the answers for your questions, right? Now let's move on to the next one. So here, let's start with this chromosomes. Have you ever wondered why do you look the same? Why do you look like your parents? Or sometimes you look like your father, sometimes you look like your mother, sometimes they say that you look, your nose looks like your grandpa, and they say that no, his ears looks like his mother, no, his eyes looks like his father. Yeah, yes. Well, yes, that's the answer. Chromosomes are here. Alright? And also not only that, they compose of this DNA, and that makes it, look like your father, mother, grandfather, etc., etc., etc. These are the hereditary materials which, pass on these characteristics from one generation to the another generation. So, you look like that. Well, now coming to this, what actually are these chromosomes? Here, it is the DNA, okay? It is the DNA, deoxyribonucleic acid molecule with part of the genetic material, that is the genome of an organism. Now, this genetic material is nothing but which contains these genes, of your parents, of your grandparents and great-grandparents. Yes, all the gene is inherited in the process, alright? And then, mostly eukaryotic chromosomes include packaging proteins which help in the packaging things, which are integral part of that, which are aided by the chaperone proteins which bind and condense to the DNA. Alright, so which binds to the DNA, and then to prevent DNA molecule and to prevent it from becoming unmanageable tangle. Suppose you just leave it like that, it just goes out, it is unmanageable, but now it becomes a great fix here. And here chromosome number is fixed in each species. Yes, it just can't go like that, suppose say you can't say, yeah, this species is 100, 200 and the next generation is having 200. No, it is fixed for each number of species. So we have this in most organisms, they occur as a homologous pair. And then each pair, the chromosome belongs to the father parent while other belongs to the mother parents. So you have this XX and XY. So you have the mother and the father, so which contribute to the child's offsprings. And then the presence of two sets of chromosome is called diploid, that is 2n is called to 46, that is in humans, alright? And presence of single set of chromosomes is called haploid, that is 23, that's also in humans, and is equal to 23, alright? 23 pairs, 46 chromosomes. So these play a very, very important role in deciding the characteristics of an organism, or suppose say human beings. Suppose say just one chromosome is missing and then it changes the entire thing, You must have seen this mentally retarded personalities, who are just born with this mental retarded. That's because the chromosome is cut off, some part of the chromosome it vanished off. And that's about this chromosomes, which plays a very, very important role in fixing up the things, alright? So here we are, thanks to the amoeba sisters for providing this, and here you have this human characteristics, that is the 46 chromosomes, alright? So you have the first chromosome looks like this, the second one like this, the third, fourth, fifth, sixth, seventh, eighth, ninth, and so there are so many of them, alright? So 23rd pair is of X and Y, which decides the sexes. So these are the 46 chromosomes which are there, alright? So this is how they look like, and each one decides a character in you. It's not like, I don't have this, no, never mind. No, each and every chromosome decides the character in you, alright? So let's go on to the cytoplasm. Now, in the cell what we have, the cytoplasm is like a jelly-like fluid which contains everything in it, alright? So if you go by the contents of the cell except the nucleus, it includes cytosol organelles, vesicles and a cytoskeleton, which contain all these things which help in the process of the cell, It is like, it's a jelly substance which contains all the things in it. And then you have this, it has two parts, that is the cytosol and the cell organelles. Now what is this cytosol? Well, the cytosol is nothing but the fluid part of the cytoplasm. And then it has a homogeneous and crystallocolloidal liquid that contains this waters, ions, proteins, all these things it contains, alright? Enzymes, vitamins, carbohydrates, lipids, proteins, etc. Everything it contains. The cytosol contains everything in it, alright? And then you have the cell organelles. So the cell organelles are submicroscopic in nature and which are specialized to perform specific cellular functions. So all the cellular functions are carried out by the cell organelles by specific. Suppose you have this mitochondria, you have this, you have this organelle, so many things, alright? So all these things are the cell organelles which are there. So like they are bounded by this membrane. In eukaryotes and also in prokaryotes, they lack membrane bound organelles, alright? So here virus lack the cytoplasm as well as membrane because it just comes and it just binds and it does its job. So this is of the cytoplasm, and cytoplasm is a very, very important role which contains all these things. Imagine if these things are not here. If the carbohydrates, amino acids, the vitamins, enzymes, lipids, proteins, if these things are not there, and what happens? The process doesn't get carried on. The cell doesn't get energy, and the cell, all things which are required are not given to them. So all these things are clubbed into the cytoplasm, alright? So now this is how the cytoplasm looks like. So you have this cytoplasm thanks to makemygf.com, and here you have this cytoplasm over here. It is, all these organelles are present in this. It is microscopic in nature. Okay, if you zoom a little bit, you'll get it. So all these organelles, everything is present in the cytoplasm. Now coming on to the next organelle, this is the endoplasmic reticulum. Now what is this endoplasmic reticulum? It can be, it can be called as the ER. Okay, it can be acronymed as ER. What this ER or the endoplasmic reticulum is nothing but a network of interconnecting membranes which are distributed throughout the cytoplasm. So in the cytoplasm, all these are distributed. Okay, so here it also plays a very important, each and every organelle which are present in the cell plays a lot of important role in functioning. Alright, you have this internal compartment that's called as the lumen, and it is separate part with the cell which is distinct protein and ion composition. That is a internal compartment. There's a lumen which is present into the endoplasmic reticulum. Alright, and the ER's folding, okay, there's a folding inside, alright, so that generates a surface area that is greater than of plasma membrane. So what is the surface area? Means the area which is present, that because of that it gives a very wide surface, alright. So at certain sites this ER membrane is continuous with the outer one and it develops a nuclear membrane. So depending on the presence and absence of the ribosomes, we have of the two types, okay. So can you guess the two types? Yes, you're right. It's a rough endoplasmic reticulum and the smooth endoplasmic reticulum. So here, what is this rough endoplasmic reticulum? It is characterized by the presence of this ribosomes, alright, on the surface. Now smooth, devoid of this ribosomes on the surface. So imagine you're catching a thread, okay, just imagine you're holding a thread, okay. There is some granules on it, and when you say it is rough, okay, so that can be considered with that. And suppose you take a smooth thread, no granules, something, it's very smooth, okay, that can be considered a smooth endoplasmic reticulum. So this endoplasmic reticulum here, it is network of interconnecting membranes. So all these things, okay, this is also important. Now let's see how it looks like. So this is how the endoplasmic reticulum looks like. So this is the rough endoplasmic reticulum, okay. So here in the rough endoplasmic reticulum you have this, ribosomes which are present here, okay. In the smooth one, they are devoid of it, okay. So here this is how this rough endoplasmic reticulum as well as the smooth endoplasmic reticulum looks like, alright. So ER, endoplasmic reticulum, this is acronym for endoplasmic reticulum, ER. So remember that, alright. And everything has got a function. Okay, if you say, so I'm here and I've also got a function. What's function? I can do so many things and all. Okay, if you say, if there's the screen also has got a function. So does this organelles have also got this functions? All right. So let's see the functions in this endoplasmic reticulum. So what are the functions? So protein synthesis is the main thing. So rough endoplasmic reticulum is the site of protein synthesis. Protein synthesis means it synthesizes proteins, it makes the proteins. Okay, that's the site in the rough endoplasmic reticulum. These proteins are synthesized, okay, and they are given to the next one. And lipid synthesis. What is the lipid synthesis? Formation of this lipid. So smooth endoplasmic reticulum helps in the lipid synthesis. Rough endoplasmic reticulum helps in the protein synthesis. Okay, two important parts. And then proteins and lipids, they are synthesized on the ear using making cell membrane. Okay, they are used for making the cell membrane. So the cell membrane is basically made by this proteins and lipids, as we saw in our previous classes. And also for the transport, this helps in the intercellular and intracellular transport. Intercellular, inside and outside the cell. So the transport is carried out by this endoplasmic reticulum. It helps in the transport. So transport of the substances, all the substances which are there inside the endoplasmic reticulum. So all the substances, whatever there, they transport through this, through this endoplasmic reticulum from one place to the another place. All right. And then you have the support also. It gives internal support to the cytoplasm. Okay. So internally it supports the cytoplasm. So everything lies on the support. Suppose I'm here, it is support of Swami. It's suppose here, and everybody supports each other. Everything supports each other. So here the support, this gives the internal support to the cytoplasm. And then smooth endoplasmic reticulum in liver cells, that takes place in detoxifying the poisons and drugs. So now you get what you take, whatever toxins you take. So when you take this, some fruits, vegetables, or whatever you eat, it does contain a little bit of toxic substances. So these toxic substances are broken down in the liver, and the smooth endoplasmic reticulum, that takes part in detoxifying many poisons and drugs. It detoxifies, okay, mainly the liver, whatever you have. So basically this alcoholics, okay, alcoholics. So what happens when they go on having, it's a toxic substance. So when they go on having, what happens? The liver is the one which gets affected. Why? Because all the toxic substances are broken down in the liver itself. All right. So let's go to the next one. So what are the differences? These are the differences between the rough endoplasmic reticulum and the smooth endoplasmic reticulum. All right. You'll be asked about the differences. All right. So mainly very, very easy. It's very easy. All right. So here it possesses ribosomes which are attached to the membrane. This is a very important point. It does not have ribosomes on its membrane. That smooth endoplasmic reticulum does not have the ribosomes. But here rough endoplasmic reticulum, it possesses ribosomes. Alright. And this is form of cisternae and a few tubules. Alright. Cisternae is inside. Alright. Inside what you do. And then you have this. This is formed of vesicles and tubules. Alright. So this rough endoplasmic reticulum participates in the synthesis of enzymes and proteins. Alright. So the enzymes and proteins, that is synthesized mainly by this rough endoplasmic reticulum. Well, on that side, glycogen, lipids and steroids, this is synthesized by the smooth endoplasmic reticulum. Alright. And also this is internal, connected with a nuclear envelope. Inside it is there. This is peripheral and may be connected to the plasma lemma. So what happens? It is peripheral in the outside side, okay, and it is connected to the plasma lemma. So the plasma lemma is one more layer. It is connected to that, and then it might develop from the nuclear envelope. Nuclear envelope is the outside one, okay. And let's like an envelope you have, that's a nuclear envelope inside outside. And then this is the smooth endoplasmic reticulum that develops from a rough endoplasmic reticulum. So this develops from this. So these are the differences to keep in mind, and very easy differences which will help you out. Alright. So coming on to the next one, this is the ribosomes. Now ribosomes, as we saw, they are present on this endoplasmic reticulum, but they have some specific function and they are known as something, okay. So here ribosomes are small rounded bodies. They are found out in this free state, so mainly on the endoplasmic reticulum. So they are found on this endoplasmic reticulum, alright. And then they are composed of this RNA proteins, ribonucleic acid. So mainly composed of this RNA and proteins. And then they are not bounded by a membrane. So there's no membrane for this ribosomes. You'll be seeing it, how they look like. And they are present in both prokaryotics and eukaryotics. Now in prokaryotics you have this 70s, 50s plus 30s. Alright. And here eukaryotes you have this 80s, 60s plus 40s. Now you must be wondering what is this S over here. Yes. S is nothing but that's a unit which is used, as we have unit for everything, like we say 1 kg of sugar, that is kg, that's a unit. Here you have this unit, there's a sweet bug, it's called a sweet bug unit, okay, sweet bug, okay, he is the scientist who found out this, okay, this unit. So that is why this is a sweet bug unit. And here that's absent in mammalian RBCs, alright. So this ribosomes are absent in mammalian RBCs and they are site of protein synthesis in the cell. So from this the protein synthesis takes place in a cell. So that is the ribosomes. So if you have a look, see, you can see the ribosomes here. So this is how the ribosomes look like, and see the protein synthesis which is going on here. Yeah, right. So that is how it looks like. See, there's a ribosomes here. So many types. So whole subunit, that's called as ribosomes, and the synthesis of proteins which goes on. Alright. So this is ribosomes. And now coming on to the next one, that is the Golgi apparatus. So Golgi apparatus also plays an important role here. Man, all the organelles play very important role. It's not like this organelle is not important, this organelle is not important. All the organelles together play a very important role in functioning of the cell. So it's like all together, as Swami says, I to we to he. So all together you get this perfect cell. Alright. So here, so Golgi complex, that's an organelle membrane, that is, it bounds sacs, tubules and vesicles. Now what are this? It secretes as biochemical components. So different biochemical components are secreted by that, and it is single complex in animal cells. Unlike the others, they are different ones. Here it is a full single complex because it contains some things. What is a complex? Which contains other things. So here it's a single complex, and then in plants they form separate units called dictosomes. In plants only. So there are separate units there. And here this membrane bound sacs, they are called as cisternae. In case of this Golgi, they are called as cisternae. Alright. And the cisternae, what it does, it occurs in sacs, tubules and tubes, vesicles, and they occur in the periphery, the outer boundary. So this is the Golgi apparatus which has all the things clubbed together in it. Alright. So this is the Golgi apparatus. See, you can see the vesicle. See, this is the Golgi body. So cisternae and all, inside this curve. So all these things are attaching. So this is a vesicle. Alright. So Golgi body which goes there. So this is Golgi apparatus inside. So this is the inside of it. Alright. So the foldings are called as a cisternae. Alright. This is a cistern, you can see. So this is how the Golgi apparatus looks like. Alright. Now functions of this Golgi apparatus. So what does this Golgi apparatus do? So that is involved in repair and synthesis of cell membranes. So always damage and repair takes place. So when the cell membrane gets damaged, it is repaired by this Golgi apparatus, and also in synthesis also. So cell membrane is synthesized by this Golgi apparatus, and lysosomes are found by this Golgi apparatus. So you saw this thing. So lysosomes, small things. So they are found by this Golgi apparatus, and all types of substance which has to be secreted, excreted, okay, are packed, okay, by this Golgi apparatus from the passage to the outside. So all this packaging thing, secreted, everything is carried out over here. It takes part in storage, modification, packaging, all this various function, all these things, it takes part in that, okay. All this biochemical produced by different components of the cell. And then you have this component of cell walls are synthesized by this Golgi apparatus once again. So any components of the cell wall, so if you take a cell wall, any components, they are synthesized by this Golgi apparatus. And complex and special sugars, they are again made by this Golgi apparatus. So see how important it is, okay. So many things, sugars, okay, cell wall, so many things, so many functions are there, alright. So This one key to how to remember the things. Just remember the main point. Suppose say Golgi apparatus involved in repair and synthesis of membrane. So you just remember it is involved in repair and synthesis cell membrane. That's it. And lysosomes are formed by Golgi apparatus. Lysosomes form. So you just need to remember the keywords which are present here. So if you remember the keywords, all these things becomes easy, very easy for you. Alright. So let's go on to the next one. This is called as the lysosomes. Alright. So these lysosomes are generally found in animal cells and they lack in plant cell. So make it a point. So this is very important. Like, you know, they are only found in the animal cells, not in the plant cells. Okay. What are these? The small spherical vesicles. Okay. Covered by a single membrane. So single membrane it covers, and they contain this digestive enzymes for intercellular digestion. So all this intercellular digestion that takes place in the lysosomes. Alright. So these enzymes are synthesized by the rough endoplasmic reticulum. Alright. So RER, rough endoplasmic reticulum. And then you have this, the damaged lysosomes burst to release these enzymes for digestion of the cellular components. Whatever has to be digested, then what happens? This cell, the lysosomes, they burst out this, the release of these enzymes which are necessary for that. Okay. And the release of, okay. They are also known as suicide bags because they destroy these foreign particles. So very important. They called as the suicide bags because they destroy this foreign particles, whatever particle comes, okay. What are this, you know, other things which are there? They are all intercellular digestion, whatever things, they are destroyed by this lysosomes. And hence, they are called as the suicide bags of the cells. So suicide bags of the cells means they destroy all this foreign particles. Alright. So remember, lysosomes, suicide bags of the cells. So once you say suicide bags of the cells, why is it called so? Because, you know, all these things it does, it digests the enzymes, intercellular digestion, all waste disposal, all these things it takes place. That's why it's called as a, what is it called as? Yes, the suicide bags of the cell. Alright. Let's go to the next one. See, this is the lysosomes looks like. Alright. See, all these things come here. All whatever toxins they come, okay, they take and they're destroyed inside. So inside it goes and they're destroyed. So that's why they call as suicide bags of the cell. So if you see all this toxin and unwanted things, they come here around, alright, they're getting inside and see automatically it vanishes off. And again they come here. So this is how the lysosomes look like. Alright. So let's go to the next one. So functions of the lysosomes are very simple. First one is the intercell destruction of foreign particles. Whatever foreign particles comes, it destroys this. Alright. And then intercellular digestion. So two cellular digestion, that's because of this lysosome. And removal of cell debris. Cell debris means all the other things which are there. Okay. The unwanted material that is removed out by this, you know, lysosomes. And then time of metamorphosis. So one person to one thing to another. So that's a time where the timing is fixed. And then you have this organs are digested by the lysosomes. So organs means whatever which is there, which is unwanted particles. So that's all are digested by this lysosome. So these are the functions of this lysosomes. Coming on to the next one, the mitochondria. Okay, a very important organelle. Okay, so if there's no power, you can't do anything. So mitochondria, when mitochondria comes, you remember, powerhouse of the cell. It gives power to the cell. Okay, power, energy to the cell. Okay, it's an energy center. All right, whatever energy, that is a powerhouse of the cell. The cell gets energy because of this mitochondria. Now, how does it look like? It's a rod-shaped cell organelles. Okay, which consists of this aerobic eukaryotes. All right, this mainly takes place in the Krebs cycle. Krebs cycle is a type of cycle. Okay, you have cycles to breaking down in metabolism. You have lots of cycles. Okay, you have this glycolysis cycle. You have this Krebs cycle. It's a type of cycle which is present in the metabolism process. All right. So commonly known as the powerhouse of the cell. It gives power to the cell. So that's why it's called as the powerhouse of the cell. All right. And they contain this enzymes which are necessary for total oxidation of food and release of high amount of energy. What is high amount of energy? The ATP. ATP, what is, when you remember this energy is given on, that is this ATP. What is it? ATP? Yes, adenosine triphosphate, right? So here, this is these produces this, mainly produces this ATP in the form of ATP. So that gives energy. Alright, more ATP, more energy, less ATP, less energy. Alright, fine. So this is bound by two membranes. One is the outer membrane, which is smooth and porous, while inner membrane is folded inside. That's called as cristae. So it's folded inside. Outer membrane is, it is developed, okay, smooth, and it is porous, which porous means it contains very small holes due to the passage of the things which has to take place, right? And this cristae contains oxysomes, which are partially embedded into the inner membrane. So here, this cristae, so you'll see how the cristae that contains this, partially embedded oxysomes which are present there. Okay, that's also a type of organelle, alright? And this mitochondria enclose a matrix having DNA, ribosomes and enzymes. So all these things are, enclosed in a matrix. In the matrix it's enclosed. So DNA and ribosomes make the mitochondria semi-autonomous. What is autonomous? Fully by themselves. Semi means partially working, alright? As they are able to manufacture some of their own proteins and enzymes, because DNA and ribosomes, they manufacture their own proteins. So here it makes them semi-autonomous. Semi-autonomous means which makes them partial functioning. Alright. So let's see how the mitochondria looks like. Here is the powerhouse of the cell which produces the power. See? So that's why you can see the power. Zoom, zoom, the power which you see. See? You see it's glowing here, right? So you can see the power which is here. So this is a cristae also, see, inner folding cristae. So you have this inner foldings, outer foldings, alright. So this is how the mitochondria looks like, alright. So since it produces the powerhouse, it's a powerhouse of the cell, right? Since you can see it's producing power, so you see the power here, alright. So coming on to the functions. So what are these functions? So the most prominent roles of mitochondria is to produce the energy currency of the cell. Mark it. Energy currency of the cell. What is this energy currency? ATP, that is, adenosine triphosphate. So this is the energy currency which provides the energy to the cells, okay? That is phosphorylation of ADP, phosphorylation, alright? So through this respiration to regulate cellular metabolism. So when you respire, as we saw in the previous class, when you respire, okay, there is this ATP formation that it regulates this cellular metabolism. And then central sets of reaction that are involved in ATP production, okay? What now this ATP production also requires something. What are the central sets? They are collectively known as the citric acid cycle or the Krebs cycle, alright? So the citric acid cycle, it is produced in the process. So that is a set of reactions, in the ATP production. So citric acid cycle or the Krebs cycle. So both are one and same. So just remember that. And then the regulation of the membrane potential. The potential of the membrane should be regulated, alright? So if that goes off, go. So it goes off, gone. So the regulation of the membrane potential is done by this. And apoptosis, that means a programmed cell death. You know that if that cell goes on, then it's very dangerous. So every cell has a death. Everything which is born has a death, alright? So here there's something called as apoptosis. So here it's a programmed cell death. The cells are programmed, not the computer program, alright? It is the cell program here, alright? So you program something, okay, the beginning and it ends off. Here they are programmed biologically, by default, alright, by nature. They are programmed, alright, at a particular time they die off and the new cells come into existence. And that's how so many cells die each and every day, so many cells are formed, alright. So here it's the regulation of cellular metabolism is also done by this. And steroid synthesis, the synthesis of this components called steroids. And this hormonal signaling, okay, so hormones are sent from one part to another. And then you have this, the sensitive and responsive to hormones, alright. So these hormones are nothing but this ductless organs. So you have this growth hormone, you have so many things, so many hormones which help with the normal functioning of the body. And mitochondria helps for the same, alright. So that is about the functions of the mitochondria. And mainly remember, mitochondria means, yes, powerhouse of the cell, alright. What is power? In case of the cell, what gives the power? Yes, ATP. So remember these things, alright. Mitochondria, powerhouse of the cell, ATP. So these things you need to remember. let's go on. Plastids. So plastids are mainly found, okay, in plant cells, only in plant cells, okay. So not in the animal sense. So they are found only in the plant cells, okay, which are specialized to synthesize and store this organic substances which are there. They synthesize and store this form. Alright. They are classified on the basis of pigment. So you must be seeing the green color, the red color flower, red color leaves sometimes. You see red color flower, white color flower, so many colors of them. Alright. So here this is, on the basis of this pigment. Alright, they form this pigment. Alright. So what is this chlorophyll? Yeah, the green pigment which is given. The green, why does plants look green? Yes, because of the chlorophyll which is present. Alright. And then you have this leucoplast, also colorless plastids, and you have this amyloplast, aleuroplast, elyplast. So all these things are there. And there's also this chromoplast. They are colored plastids, green and non-green plastids. So all this plays a very, very important role. These plastids play an important role because they give the color. Alright. Now next, so this is how the plastids look like. Alright, that's a process which comes here. Okay, so the stroma, and they have the thylakoids. It comes, and see, this is how it looks like. So going on to the differences between the leucoplasts and the chromoplasts. Alright, so leucoplasts are colorless. This chromoplasts are brown or reddish in color. And then you have this leucoplasts are cylindrical or round in shape. Chromoplasts are irregular in shape. And then these are found in unexposed cells, the leucoplasts. These are found in exposed and unexposed cells, both. And this take part in storage of food, this leucoplasts. And chromoplasts do not store food, do not take place in storage of food. And then they can change to other types. Here they provide coloration to the organs. So these are the main differences between the leucoplasts and the chloroplasts. Alright. So coming on to the next one. You have, let's go to the chloroplasts now. So we saw what is chloroplast. So this is bound by two membranes. Inside the chloroplast, it's a clearly marked. Alright. So a colorless matrix called stroma and a membranous system called grana. So stroma and grana are present in this chloroplasts. Alright. So each granum that consists of stack of membrane bound flattened sacs. A sacs, which is what a sac? Something which you carry. So they are flattened sacs, okay, called as thylakoids. So these are thylakoids which are the flattened sacs. And then these pigments like chlorophyll, carotenoids and phycobilins. So they contains this thylakoid pigments. So this is called as chlorophyll, carotenoids. This all are called as this thylakoid pigments. Alright. And then the stroma also contains this enzymes for dark reaction. Alright. So dark reaction which takes place in the cell. So you have this DNA, RNA and ribosomes also here. They make this chloroplast semi-autonomous. So semi-autonomous, partially, they prepare their own food, okay. And then this is also known as the kitchen of the plant cell. So chloroplasts are known as the kitchen of the plant cell. So in the plant cell, everything, all the food and everything is prepared. So what is the kitchen? Yummy. You have all these things in the kitchen, right? So you have food, everything is prepared in the kitchen. So here, in case of this plant cell, this chloroplasts are known as the kitchen of the plant cell. Alright. So that's about the chloroplasts. Alright. This also give the green pigmentation which contains chlorophyll. Alright. So these are the chloroplasts. So see, so this is how they're moving. It's a live cell image which is taken. So you can see the chloroplasts here. So if you see the structure, this is the outer membrane which is here. This is the inner membrane which contains the thylakoids, okay. As I told you, the stroma, right, which interconnects this board. So this is a thylakoid and there's a stroma over here. And then there's an intermembrane space, means there's a space between two membranes. And you have this granum. This is a granum, stack of thylakoids. So one thylakoid, this is one thylakoid. So stack of thylakoids, that's called as a granum. Alright. So this is the figure of the plant cell, the chlorophyll structure. And if you see it in microscope, if you see it in electron microscope, you see this is the structure which goes on. So that's about the chloroplast. And then let's go on to the functions of this chloroplast. Alright. So the most important function is synthesize food. by process of photosynthesis. So in the previous classes you saw what is photosynthesis. Synthesis of the food by the plant. So this chloroplast, this is the most important. Chloroplasts are, they help to synthesize the food by this process of photosynthesis. Photosynthesis because of the light, the sunlight which comes, that synthesizes the food. Alright, and this absorbs light energy and converts into the chemical energy. So sunlight energy which you get, that absorbs it and that converts into the chemical energy. And this chloroplast has a structure called chlorophyll, okay, which functions by trapping the solar energy. Alright, chlorophyll also is important for giving the green pigmentation in leaves. Anything, the leaves, everything, the plants look green because of this pigment. Okay, so you have this chlorophyll pigment, and in the food, in all green plants, so all these green plants, chloroplast, chlorophyll is present in all these green plants, and they also produce this NADPH, okay, and the molecular oxygen by photolysis of water. Photolysis, when water is that lysis breaking down, when water comes, so it breaks down that. All right, and then it produces this ATP, adenosine triphosphate. So that's the process of photosynthesis. In this process of photosynthesis, if you have seen the previous classes, you learn what is photosynthesis. What is photosynthesis? Preparation of the food, okay, by the plants using the sunlight. That is called as, photosynthesis. Synthesis, preparation, photo, light. So preparation of the food by the light. And then the carbon dioxide that is obtained from the air, that is used to generate this carbon and sugar during the Calvin cycle, okay, or the dark cycle reaction of the photosynthesis. So this is the carbon dioxide obtained from the air. So that is also what happens during the process of photosynthesis. Okay, there's a generation of this. How? Because it uses the light and it generates the food. All right, in the presence of carbon dioxide. Okay, it takes in carbon dioxide and gives back the oxygen. All right, so that's about this. So next, let's go on to the vacuoles. All right, so the vacuoles, what is vacuoles? These are membrane bound non-cytoplasmic sacs. Okay, these are, they have this membrane. They're non-cytoplasmic, so they have this membrane bound. Okay, that contain non-living solid or liquid contents. Now, to see, there are these three types, okay, sap vacuoles, food vacuoles, and contractile vacuoles. So these three types are present, three types of vacuoles are present here. Now coming to one by one. So the covering membrane of the sap vacuole, that's called as tonoplast. So vacuole, sap vacuole, okay, inside it is, so the covering membrane, that's called as a tonoplast. And then the fluid filled content of the vacuole, that's called as sap or tonoplast. Alright. So recently digestive enzymes are reported to have this vacuoles, okay, vacuoles of the plant cell. Okay, why? Because we have this plant substance cell. So that recently this digestive enzymes are reported in vacuole of the plant cells. And then functions, here only we will study the functions. What are the main functions of this vacuoles? Alright, it is maintaining this osmotic pressure, storing toxic byproducts, and providing turgidity to the cell. So this osmotic pressure, the water, what is required, okay, that is maintained by this, by this vacuoles. And then storing the toxic products. So toxic byproducts, what are the byproducts which are produced by these toxins? So that it stores that. And turgidity to the cell. So maintain the rigid turgidity to the cell. Alright. So that is about this vacuoles. So this is how the vacuoles look like. Okay, this is a paramecium vacuole. Alright. So here it's produced like this. This is electron microscopic image, okay, of the paramecium vacuole. So if you see the organism paramecium, this is the vacuole which is present here. And now coming on to this peroxisomes. So what is peroxisomes? They are small vesicles, alright, which are single membrane bound organelles. Where are they found? Yes, they are found around the eukaryotic cells. So they are found around these eukaryotic cells, okay, and they have also got a specific function. What do they contain? This digestive enzyme for breaking down this toxic materials of the cell, alright, and oxidative enzymes for metabolic activity, alright. So this helps mainly in the metabolic activity. So break down this digestive enzymes, okay. So here the digestive enzymes they contain and they break down this toxic materials in the cell, alright. And here they are heterogeneous group of organelles. And how they are they are in the presence of this marker. What are this marker enzymes which mark to distinguish from the other cell organelles? Means they mark the other cell organelles. So you have this, suppose you want something, you mark there, okay, which distinguishes it from one part to the another part. So these are present in that. And this peroxisomes plays a very important role in lipid production. Okay, they are involved in conversion of reactive oxygen species such as this hydrogen peroxide into safer molecules like water and oxygen. So all these things, so if you take a component or this toxic substances, it breaks down and then it makes it into safer substances like molecules like water and oxygen. If you take this hydrogen peroxide, okay, it breaks down into water and oxygen. All right. So these are the peroxisomes, the small ones. They are not, so these glowing ones, if you can see, they are called, they are colored glowing ones. So these are the peroxisomes. All right. So now functions of this. So synthesis of this ether glycerolipids and plasmalogens. So these are the components which are present in cells. So they are the synthesis, the making over of this glycerolipids and plasmalogens are done by this thing. And the formation of bile acids, dolicol and cholesterol, so that is carried on by these peroxisomes. And then the catabolism of purines, polyamides and amino acids. Catabolism. What is metabolism? What is catabolism? Yeah, forming up and breaking down, right? So here you have this catabolism of purines, polyamides and amino acids and the detoxification of reactive oxygen species. Okay, detoxification. So the toxins are removed from this oxygen specific reactive oxygen species. All right, and in metallotrophic yeasts, okay, this is a type of yeast. The peroxisomes are also involved in the metabolism of methanol and methyl amines. So these two compounds, thus, peroxisomes are involved. So these are some of the functions, important functions of this peroxisomes. All right. So all you can remember the synthesis of ether glucose and plasmalogens. Okay. Formation bile acids and dolicol and cholesterol. So catabolism, purine. So if you remember this key points, all right, if you remember this, note down this key points, you can remember it more and more nice. So coming on to the next one, that's the centrosome. So the centrosome, it is a small naked organelle present near the nucleus of the cells. So near the nucleus you will find the centrosome. Okay. So and some in the lower plants. Okay. Cells and some animals have it in the lower plants. So here it is present near the nucleus, and then it has a sheath of special cytoplasm called centrosphere. This is the special sheath of cytoplasm. All right. So that is called, it is called as a centrosphere. And then it is enclosed by two microcylinders called centrioles. So two microcylinders, it has got centrioles. So you see two sides of cylinders, that's called as centrioles. And then these centrioles are like right angles to each other. What's the right angle? This right angle. So they are right angles to each other. And it has 9 triplet fibrils, okay, obliquely on the periphery outside. All right, 9 triplet fibrils. So 3 into 9. So that many of that has. They form this astral spindle fibers. Okay, this astral spindle fibers are nothing but you have these lines which are projecting. Okay, they are fibers and produces basal bodies that form cilia and flagella. Now the cilia and flagella are too important. Okay, what does this? They help in the locomotion. The cilia and flagella help in the locomotion and the absorption. So this is about this centrosome. All right. So this is how the centrosome looks like. See, it's here, and thanks to makemygf.com, and you have the centrosome. This is how it looks like in the electron microscope. And if you see the diagram, so you have this is the distal appendages, okay, subdistal appendages over here. This is the distal ends, this is the proximal ends and interconnecting fibers. So these two are held by the interconnecting fibers and they are at a right angle. So if you see they are at a right angle. And this the microtubule triplets. So triplets means 3 together. So microtubules, these are the microtubules, small one. How are they? 3 together. If you see, all 3 are clubbed together. All right. This is the daughter centriole, okay, daughter centriole, and this is a mother centriole. So here, from here it forms. So this is how the centrosome looks like. All right. So now the functions of this centrosome. So it assists in the cell division during the mitosis. So cell, it divides. So it is very important the cell divides, okay? In the mitosis, meiosis, there are two types of that, mitosis and meiosis, okay? Mitosis, it is a protection where cell divides, okay, into 1, 2, 2, 4, 6, and it goes on dividing like that, alright? So here they help in direct movements of microtubules and cytokeratin structures. So they help in the, what is this, direct movement of this microtubules, and also facilitating the change of shapes of membrane of this animal cell. The shapes of these membranes are doing, they facilitate for the shapes, alright? So changes in shapes. Also they play important role in mitosis. How? By organizing the mitotubules, alright? So in the when there is a division, they organize this microtubules, alright? The centrosomes are distributed each to the daughter cell. So if you saw that, okay, they're distributed to the each daughter cell. And then they also stimulate the changes in the shape of cell membrane. How? By the phagocytosis. What is this phagocytosis? Inside. So eating of the cells, okay? So phagocytosis, alright. So here they maintain the chromosome number during the cell division. So this is very important, the maintaining of the chromosome number during cell division. So when the cell division takes place, when the cell is dividing from one part to another part, the maintaining of the cell, chromosome is very important. Suppose if one chromosome gets mixed, the missed off, then gone. The entire process goes off, alright. So here they maintain thus the chromosome number during the cell division. Alright, going on to the next one. What is the difference between the plant cell and the animal cell? Alright, so you've seen the plant cell, you have seen all the organelles. Now let's see the differences between the plant cell and the animal cell. Here, the cell wall is present in the plant cell. In the animal cell, it is absent. Okay, the cell wall, you saw what is the cell wall. And plastids are present. Plastids are absent in the animal cell. Here, centrioles and centrosomes are absent. Here, centrioles and centrosomes are present in the animal cell. And vacuoles, large and less. Vacuoles, small and many. Alright, here you have the large vacuole. Here is very small ones. And then nucleus lie on one side. Here, nucleus lies at the center over here. And cell is rigid here. Cell is little bit flexible. So that's this rigidity gives plant cell, that's why it's very rigid. So animal cell, it is flexible. Right. So Golgi contains dictyosomes, okay, here in the form of vesicles. It contains here vesicles. And this is larger in size, this is smaller in size. Alright. Starch is stored in the plant cell. Here glycogen is stored in the animal cell. So these are the brief differences between the plant cell and the animal cell. Now let's see. So test your skills on this, alright? On this part you can test your skills. So first one, explain chromosomes along with haploid and diploid. So go to the chromosome slide and see what is the haploid and diploid there, okay? And describe cytoplasm and its functions. Go to cytoplasm and see what all it does, how it looks like, do that. And mention the functions of the endoplasmic reticulum. Go to the endoplasmic reticulum, remember the functions, how it looks like, what is looks like, okay? Now, what are ribosomes and where are they found? So what are these ribosomes, what do you tell and all? So go back to that and then remember it and you can write it down. And how does the Golgi apparatus work? So there's the working of this Golgi apparatus, how does it work and how does it go forward? Remember that, alright? Which cell organelle is known as a powerhouse of the cell? I told you about the power, okay, ATP is power. Which cell organelle is known that? So you just remember that, okay? You can write it down, alright? So describe this organelle which is termed as a suicide bag of the cell. So I also told you what is the suicide bag of the cell, okay? Go back to the slides, previous slides, you'll find the answers. And then what are the functions of centromere, peroxisomes and the chloroplast? So all these functions, okay, you go back to the slides and find out centromere, peroxisomes and chloroplasts, okay? You find the functions of this. And let's sort the differences between plant and animal cells. So these are the things, test your skills on that, alright, and then you'll come to know about that, where you stand. Go to the previous slides, okay, evaluate for yourself. If you get it wrong, never mind, go back once again, okay, learn the things once again and then you correct it, and there will be a point of time where you'll get everything correct, and that's where you can tell, yes, I've understood, alright. You can write it in your own words, you can term it in your own words, but understand the concept, alright. So this marks the end of the chapter and also of the third thing. Thank you. Sai Ram, Swami bless you.